Thank you so much for having me here, and thank you all for coming to see a full room for a biochemistry research lecture. Is quite a sight. I want to say at the beginning that I'm happy to make all of my slides available, so you don't have to worry, but you are welcome to photograph them if you so choose to. So today what I would like to do is present one possible theory of how acupuncture works on a biochemical basis. I want to frame it to say that I don't believe that this is the only way that acupuncture is working biochemically, nor do I think that acupuncture, as we practice it, can be fully explained only looking at biochemistry. That said, I think it's useful to be able to, when you need to, depending on who you're speaking with, and also just quite cool and interesting. So we'll do a background of the key concepts, and usually I try to explain things in a way that a 4-year-old could understand. However, for this subject, using some terminology is fairly unavoidable, so I will do my best to explain it to make it clear, but this will allow you to use the actual words and to find it in the research as it will be referred to. But feel free to raise your hand if I'm being confusing or if I haven't explained things clearly. So the advantages really are about having an ability to speak about acupuncture using the dominant paradigm is just a useful thing to be able to do. And I think that being able to do that actually can improve our clinical outcomes, which is the whole point of the exercise in the first place, and as well as communicating our confidence when we're speaking with patients and medical professionals. Oh, yes, we're going to go forwards, not backwards. And not that we always need to speak to our critics, but it can be helpful sometimes when necessary. And this particular model that I'm going to present particularly is a good argument against two of the critiques that we get from skeptics. One is that acupuncture lacks biological plausibility. And also that it's somewhat dubious. We claim to treat everything from infertility to asthma to anxiety, and that in itself is suspect. So this model gives a very clear rational explanation of how that can be and is entirely possible to do that. So first, to give a little background, because I think it's interesting to understand stories and also to understand how it is that we know things, and really how a lot of our information from the modern biological perspective is actually fairly recent. So the neuron was only discovered in 1887 by this wonderful guy who really, I think, was a very synthetic thinker. So he was a neurologist, but also a beautiful artist. And he writes about the moment of discovery, saying that against a clear background stood black threadlets, some slender and smooth, some thick and thorny, all was sharp as a sketch with Chinese ink on transparent Japanese paper. A look was enough, dumbfounded. I could not take my eye from the microscope. I was like, it never occurred to me that there was a time not that long ago we didn't actually even know that we had neurons. And this is, I think, really impressive. This is the drawing that he drew of a hippocampal neuron, and this is from a modern electron microscope. So I just think that's quite cool. So then there was this question. They discovered that the brain had these separate cells and that there was a space between them. So the next question became, how does the signal get from one to the other? And part of that was answered by this guy, who the answer came to him in a dream, that he literally dreamt one day and he imagined this experiment with these two frogs. And so he took one of the frogs and stimulated its vagus nerve. Don't remember the details. And it was in a pool of some sort of liquid, and then he kind of took that frog out and put another frog in and realized that he was able to have a similar nervous system effect, that there was something in the pool from the first frog. So he basically discovered acetylcholine. And this actually then, this period initiates this kind of time that's referred to as the War of the Soup and the Sparks, which I bring up because I feel like there's a lot of similarities with kind of where we fit into modern medicine, where we're proposing a certain model of how things work that's going against the dominant model. And in this case, it was the kind of lowly pharmacists who were proposing that it's biochemical in nature, and then we got kind of lab guys with their fancy tools and fancy toys that were proposing electricity. But it turns out that the soups were right, that signals go from one nerve to another via biochemistry. And so this will be familiar to most of us, that we have this, the two parts of the autonomic nervous system, the automatic nervous system. We've got the sympathetic fight or flight part of the nervous system, and the parasympathetic rest and digest part of the nervous system that we're trying to help encourage our patients to stimulate more, to stimulate the sympathetic less. And so that's what these two are called, the adrenergic and the cholinergic, or acetylcholine nervous system. But, and that was the story. And actually that's really the main story that most people know, that that's the end of it, and even in conventional medicine. But this guy from Australia then does an experiment. I don't believe this came to him in a dream. This one was guinea pigs. And he did an experiment where he was able to completely block the sympathetic and the parasympathetic nervous systems, but signals still got through, which suggested that there was another mode of communication aside from those. And he was able to deduce that it was actually through something called purinergic signaling. So signaling with purines is what he discovered. And just to kind of demystify, a purine is a type of molecule that has this double ring structure. That's what we're referring to. And there are quite a number of purines, but specifically when we're talking about purinergic signaling, and we'll get to acupuncture, we're talking about ATP, which a lot of you will be familiar with as the main energy currency in the body, and adenosine, which is basically ATP without 3 phosphates. Are we all still here? We're still okay? Okay, good. And then just to give, this, to me, everything in biochemistry, I was just talking about this earlier, I'm so grateful that I did my Chinese medicine training before studying biochemistry, because all of biochemistry is yin-yang theory, and it makes it incredibly easy to learn. And I felt so bad for medical students because they learn all this information as if it's completely discrete with no pattern to it. Just learn yin-yang theory first, and then it's fine. So you can see this yin-yang dynamic in this sort of this continual cycle of self-regulation of these levels. So this theory was completely resisted for about 20 years, but then in the 1990s, when researchers were able to clone the receptors for these purines, it was finally accepted. It is now established that every nerve uses ATP as a co-transmitter. So it's kind of important, right? And also, I kind of will suggest, so it's important, but also purines and purinergic signaling and ATP seem to exist almost with like the earliest forms of life on the planet. So that makes me think it's even more important. Like everything alive has this system. So what is it? So with any signal in the body, particularly hormones, neurotransmitters, we have the release of the signal, we have the receiving of the signal, and the inactivation. I think of the metaphor of phoning someone, right? So you phone the person, the phone rings, that's the kind of signal being received, and then once you pick it up, hopefully the phone stops ringing. So that's what we're talking about here. So how do purines get released? Well, they get released if tissue gets stretched or deformed. Shear stress, like blood flowing through an artery, heat, stretch, swelling, if there's deficient oxygen, or damage in dying cells. And so if you are thinking that some of these seem familiar to you, then there's good reason, right? That this is how we initiate purinergic signaling. And this is a picture from a study that was done a few years ago, and you can see this acupuncture-like looking probe that they use, and those red dots are ATP. So the probe goes in, it kind of damages the cell, and then you can see over time the ATP coming out. And then this is looking at ATP being released with mechanical stretch, so stretch of tissue at 21%, and you can see these like kind of beautiful ATP clouds coming up, right? So we can kind of see what that looks like. So, and then when ATP is released, it's quickly getting converted into adenosine. So we've got the release, then we've got the reception of the signal, and what's kind of cool to pay attention to here is that the action of something in the body is actually really more to do with the receptor than the thing itself. So as an example, serotonin can either speed up the transit time through the gut or slow it down, depending on the receptor that it attaches to. So the same thing with ATP. It can be anti-inflammatory, it can be pro-inflammatory, it can be a signaling molecule for acute pain or for chronic pain, depending on the receptor type that it's binding to. There are receptors for it in all tissues and all body systems, which is unusual for anything in the body. And so I'm now kind of putting this here, not to scare you, but only because these are the things that come up if you look at any research of acupuncture and its effect on purinergic signaling or ATP or adenosine, just to be aware that they have these names, like adenosine A1 receptor. There will be no quiz on these functions. So there with that. And then we have the inactivation, and the inactivation is really just a conversion of yang into yin, really. And then this is just a schematic that kind of shows that ATP loses one of its phosphates, becomes ATP, then AMP, then adenosine, and you've got this like water cycle thing going on. So you might be asking yourself, what on earth does this have to do with acupuncture? But hopefully if my setup was clear, you have a good idea of what it does. And what's interesting is that the same Australian dude who discovered purinergic signaling in the first place was the first person to come up with the hypothesis that acupuncture might be working via purinergic signaling. So this is, some in the nastier moments of our skeptics, we're accused of co-opting other ideas, but this actually came from the dude who discovered it. He suggested that acupuncture might be working this way, and he says that perhaps poking and stretching by needles and application of heat leads to release of large amounts of ATP from fibroblasts, which are stimulating tissue repair. And then the ATP then binds to the receptors on the nerve endings, and then perhaps this transmits a signal to the brain and the nervous system, and then perhaps that regulates the higher functions, and we get an acupuncture effect. And this was one of the schematics in his paper. And this is one that I redid, which, depending on your visual preferences, may be simpler, but it's just, it is a clear way of explaining one thing that we know, that when we put a needle in, that happens, that can explain acupuncture's varied effects. So this was a theory, and then it was tested first in mice. And what these guys did was they had regular mice and then what they call knockout mice. And they had a group of mice where they knocked out the gene for the receptor for adenosine. And then they gave them acupuncture. And they found that adenosine is released during acupuncture 24-fold. And that ATP returns to baseline, but adenosine stays elevated. And that if mice did not have receptors for adenosine, they did not get pain relief from acupuncture. And I do want to say that when I communicate with critics and skeptics, it's not normally my aim to convert them, because it's usually like a fairly religious sort of level. It's usually just to present a counter argument for somebody who might be watching. But in this case, I actually presented this on a skeptic's website and converted a skeptic by mentioning this study and explaining the design. So these things are important. It's not just this study happened and it showed this. It's like giving a clear visual to the person of why this is not belief and attention and placebo. Like, look at it. So something to keep in mind, that it's good to be aware of these types of studies. And the authors wrote, although acupuncture has been practiced for over 4,000 years, it has been difficult to establish its biological basis. Our findings indicate that adenosine is central to the mechanistic actions of acupuncture, which is very cool. But it doesn't quite do what it needs to do unless it's a study in humans. And so this study was then done in humans in 2012, and they confirmed the findings. And they used stomach 36, and they found that the extracellular adenosine increased significantly, and they used a control point that was equidistant from the probe, and they did not have a rise in adenosine. like point specificity, we like it. And also that it was not observed without needle rotation, which is interesting. So, we respect all styles, but this was an interesting finding that it really was the winding of the tissues around the needle that was stretching the fibroblasts that was causing it to granulate for what it's worth. And so, sorry, I'm going backwards. Haven't we seen that slide? And so they write, our results indicate that acupuncture triggers adenosine release in humans comparable to that previously observed in mice. Interestingly, acupuncture needle placement in a non-acupuncture point in equal distance to the microdialysis probe did not trigger significant increases in local adenosine concentrations. So there you go. Now, I have been surprised to observe that when I've presented this study, people get really upset. Like I'm trying to take away their coffee, or their patient's coffee. As was mentioned, I'm also trained in nutrition and functional medicine, and I am not pro or anti-coffee in general. So I think that's ridiculous. But I do think that caffeine, being the world's favorite purine, is in close communication with the same system that acupuncture is using, for what it's worth. Personally, I now schedule my acupuncture appointments for myself in the afternoon, and I have my morning coffee. So, what I want to suggest, I can't prove this, but this is just where my thoughts are going, is that this diagram showing all the different cell types that are being affected when we needle, then triggering the release of all these different chemicals, I'm seeing research emerge that suggests possibly that ATP and adenosine are kind of up here, and that everything happens downstream. That's my reading of things thus far. And then even more recently, this study came out where they were looking at the roles of adenosine and ATP and histamine, and they did every possible combination of knocking out genes and giving chemicals to enhance adenosine, to enhance histamine, to block adenosine, to block histamine, to really fine-tune what the effect is and what's happening. And this is a little bit of a confusing graph, but these are the controls. These are the guys who are in pain. Acupuncture helps get them back out of pain, but enhanced acupuncture and enhancing adenosine gets them out of pain. Histamine, less so. So it seems that histamine is important for acupuncture's effect, but again, it seems to be downstream of adenosine and ATP. So it seems to create this sort of domino effect. And so, some other things, because I've been interested in this question of how acupuncture works from a biochemical basis for quite a long time. So, I have all these papers that I'm used to, and one of them was looking at nitric oxide and something called a calcitonin gene-related peptide, which is particularly relevant to your migraine patients, right? And so that acupuncture seems to affect these things. But when I went back to read the study, it said that, interestingly, binding adenosine phosphate compounds to their receptors on vascular endothelium cells induces nitric oxide production. So acupuncture is good because it increases nitric oxide, which relaxes the blood vessels and has a pain-relieving effect, but it happens once ATP binds to it, which is kind of cool. And then we see that something similar with this pathway of something called brain-derived neurotrophic factor, which is like miracle grow for your brain cells. You want more of that. That's a good thing to have. And then again, looking at this, going back to look at that paper, I see acupuncture can inhibit neurodegeneration via BDNF. Moreover, recent studies have reported that acupuncture can increase ATP levels at local points. We've also demonstrated that acupuncture could activate monocytes, which are a certain type of immune cell, and increase the expression of BDNF via the stimulation of ATP. We're starting to see a little bit of a theme, which is kind of cool. And lest we forget, I will mention that there's also some evidence that moxibustion will induce the release of ATP as well, and which makes sense because we saw that heat would do it. So, what clinical areas could this possibly have any relevance for? Well, I was amazed when I started looking at this. So, first, Bernstock and his colleague say cholinergic transmission is unique when compared with other systems, utilizing extracellular chemical transmitter molecules in its pluripotency and omnipresence. I love that sentence. So normally, when we look at the body from a reductionist perspective, we see a high degree of compartmentalization, but not so with this system. Most transmitter systems are anatomically segregated, having a preferential manifestation in certain systems and tissues. That said, in contrast, ATP and adenosine are produced and released by cells in all tissues. And most cells, no matter how different their phenotypes are, express purine receptors, whereas purinergic signaling contributes to a wide variety of physiological functions, so it might have relevance in a lot of areas. But then again, it's fine for an acupuncturist to say that, right? But this study was in Trends in Pharmacological Sciences, so that, maybe there's something to it. And they write this title of this is Adenosine as a Multi-Signaling Guardian Angel in Human Diseases. And they write, the importance of adenosine for human health cannot be overstated. Indeed, this ubiquitous nucleoside is an integral component of ATP and regulates the function of every tissue and organ in the body. So that seems kind of important. And so, of course, we'll just make lots of drugs to mess with the system and hope nothing goes wrong, and it'll be great. So then, or you could go for acupuncture, of course. So, looking at a few examples, if we look at pain as a clinical area, we see multiple purine receptor subtypes are involved in pain pathways both as an initiator and as a modulator. So again, it's like fine-tuning this entire system. Some receptors contribute to acute pain, other receptors contribute to chronic pain associated with nerve injury or chronic inflammation, and yet others contribute to neuropathic pain. So all these different types of pain, we have the same signal, and depending on what it binds to, creates different types of pain, which are probably beautifully described with the terminology that we use. But actually, it just occurred to me, wouldn't it be interesting to see if we could look at different B syndromes or different types of pain and see what these levels are. Okay, headache and migraine. So there's a review on the role of adenosine signaling in headache. And they say clinical evidence and rodent studies have suggested that adenosine signaling also plays a critical role in migraine headache. In this review, we highlight evidence that supports the involvement of adenosine signaling in different forms of headache, headache triggers, and basic headache physiology. This evidence suggests that adenosine A2A receptors, which you're now all familiar with from that previous scary slide, as a critical adenosine receptor subtype involved in headache pain. So this molecule brings together all different types of headaches, which is kind of interesting. And adenosine is involved in a number of the common headache triggers and may act as a unifying mediator of like all of your seemingly random triggers like stress or weekend and holiday or coffee or chocolate or red wine or cheese may all mess with adenosine or ATP. And further lending support to this, people who have genetically wonky adenosine A2A receptors may be more susceptible to migraine with aura. So that makes that point. Okay, now looking at the immune system, this, when I saw, because I'm doing research with this, I'm like looking at the title of this, I'm like, what, am I hallucinating? Extracellular ATP and adenosine, the yin and yang, and immune responses, question mark. And then I like Googled these guys, and they're not even Chinese medical people. Right? But they see what's going on here, that it's a balancing act, that the same signal can have different effects depending on what it's doing. And so here you can see that ATP is stimulating these immune cells to create certain things. Adenosine, which is the yin-yang pair in this kind of mutually regulatory dance, is inhibiting those same ones and increasing Treg. So maybe it's not such a good idea to mess with the system pharmacologically, perhaps. One might say. And then, of course, if it's affecting the immune system, it's going to have an effect on cancer. So we look at targeting adenosine in cancer immunotherapy, a review of recent progress. The adenosine pathway plays a key role in modulating immune responses in health and in disease. In health, anti-inflammatory effects of adenosine balance pro-inflammatory ATP, limiting tissue destruction by activated immune cells. In disease, this balance is disturbed. Recent studies focusing on molecular and cellular mechanisms associated with adenosine effects on tumor progression suggested that adenosine might serve as a potential immune checkpoint in mediating cancer-associated immune suppression. So I can, if you've looked into the immune system in any detail, you'll know that it's quite complex, and there are a lot of players so that are kind of dancing with each other. But what they're suggesting is this balance or imbalance that serves as the checkpoint to whether or not a cell becomes tumorous. So that seems kind of central. If we look at the cardiovascular system, we can see that purinergic signaling shows up again. So this review is looking at the different cell types and then the different receptors that they have and what happens when those two meet. And actually, what I've come to realize is that if you've ever kind of thought about like, how does a person sort of know that their heart is beating at an appropriate thing, because it's like the background environment doesn't change too much. It's ATP bonding to receptors. That's the temperature reading that the brain is getting, and it's when the blood goes through the blood vessels and creates that shear force, that is then releasing a certain amount of ATP or adenosine, and that's the signal that's going back to the brain. And so it is in the gut. So it influences things like intestinal motility, secretion, absorption, reflexes, and the mechanosensory transduction reversible organs, how we know when we need to poop, for example. Okay, so now that would be a kind of thorough look at, so we've made the point. So acupuncture is going to stimulate the release of ATP and adenosine, and that kind of accounts. We can trace that from beginning to end, and it accounts for its wide effects. The problem is that it's been about 25 minutes, and I was given 90 minutes to talk. So I was like, okay, well, what else am I going to say about this? So I had a think. I thought, well, this is like kind of my pet theory, and then I thought, okay, no, Mel, this is your pet theory. Step back. What's the best other theory of how acupuncture works, and what the channels are? So which I will show you, and I thought, okay. Is it related to purines? And the answer, yes. So we can keep going. So what does that have to do with how, the channel system? So everything I presented was on a microscopic level. We still don't have any sense of the channel system or points or distribution. So we need to come back to this question of how do organisms organize? And so there's some really cool literature from this guy here. He may have a clone. He publishes like 300 papers a year. But he talks about top-down models. And this is really one of the, maybe this is obvious, so I apologize, but one of the fundamental differences with how we view the human body and health versus how conventional medicine does is that they're really stuck on these bottom-up models that, if you have one single gene switch that's going to change the entire organism, yeah, that happens, but that's the exception, not the rule. And the same thing, that's how all medication works. All medication is based on this bottom-up way of looking at things. If we just change the concentration of this chemical, it's going to change everything up here. Problem is it does a lot of other things besides what you're trying to do because it is bottom-up. So if we look at what Michael Levin says about top-down models, it is widely assumed in developmental biology and bioengineering that optimal understanding and control of complex living systems follows from models of molecular events, right? This bottom-up model. The success of reductionism has overshadowed attempts at top-down models and control policies in biological systems. However, other fields, including physics, engineering, and neuroscience, have successfully used the explanations and models at higher levels of organization. Exploiting the dynamic regulation of pattern formation and embryogenesis and regeneration requires new approaches to understand how cells cooperate towards large-scale anatomical goal states. How are we doing? Are we still good? Yeah, okay. And so, a way to sum this up is that all of molecular biology can be summed up in this sentence from James Watson, who commonly takes credit for discovering the double helix, which is that DNA makes RNA, which goes to protein. That's all of it. That's biochemistry, right? But there's a problem, which is that we start off as a single cell, and then we usually end up like looking something like that, right, in three-dimensional space. And the central dogma of biochemistry, while cool and good at explaining lots of things, doesn't explain how that happens. That's like a big thing to be missing in your model of how you're treating people, right? So every single cell in the body has identical DNA information in it. And if you start asking someone, like, they will tell you a bit about, oh, well, it knows it's a liver cell because of, the environment, but it does not tell you how this knows where to put a head, where to put the feet, left, right, front, back. And this is another way, to look at that we start off as a single cell, but then at some point we have a front and a back, and then we have a top and a bottom, and then we have organs, and that is not explained in the central dogma. So that's a pretty big gap. And so interestingly, what I learned recently, is that what we're describing here, this three-dimensional patterning, is called morphogenesis. The first guy who actually came up with this model is, what's his first name? Alan Turing. So the code breaker from World War II, from Bletchley Park, actually came up with this using mathematical modeling. How cool is that? So what we see is that this is, I think, so it's an embryo, but what we're looking at here are something called morphogens, okay? And the way these work is by creating a concentration that diffuses, that allows cells to know where they are in three-dimensional space, so that you know if you're going to become a head or a tail, or left or right, etc. And here's another view. This is looking at C. elegans, a worm. So if we're just looking at the conventional view of how we look at things, then we just see cell, two identical cells. Actually they're not identical. There's a front and a back, right? And so we've got the morphogens there, and that's part of what is telling everything where it is in three-dimensional space. And again, this is, I didn't know the xenopus. I had to Google image that. It's like a frog-like thing. So again, that we get the cell division, and the cells could be identical, but they're not. And it's from this morphogenic gradient that we get differentiation into ectoderm, mesoderm, and endoderm. And there's a strong electrical component. So with that, bioelectric fields have been shown to interact with morphogens and guide growth control. The morphogenetic singularity theory, published a decade ago, suggests that organizing centers have high density of gap junctions. So these are the centers that are releasing the morphogens in the body, and high electrical conductance. They are the singular points in morphogen gradient and bioelectric field. So chemical and electrical, yeah? A growth control system originates from a network of organizing centers containing under-differentiated cells and retains its regulatory functions after embryogenesis. In other words, this is the system that allows the embryo to develop and know top from bottom and left and right and where to put organs, and we don't get rid of it after that's done. That's how we regenerate our tissue, and that's how we regulate our organs. Oh no, I've offended her. She told me she had a train to catch. And so, in this paper, he shows that this was the theory, and that actually we can see this in this cell called a blastopore has this electrical gradient. And this was back in 2001, but now we're in 2019. So what have we learned?
What we're able to do now, is now possible before, is combine molecular biology with these understandings of the physiology, the electrophysiology that's going on, so that we can really control it at the genetic level.
I love the caption of an electric phase. It's a really perfect way to explain what it is that you're seeing in that video. So we're actually able to see where cells are acting as a battery where they're accumulating negative charges. And the amazing part was that those negative charges overlap in areas where those cells are going to become parts of the phase.
So just cell divisions, lots of activity, front end of spinal cord, there's the brain, mouth, pharyngeal arches, eye, nose, ear.
All of these things are in some way being regulated by a single type of signal. That is just beautiful to me.
The finding suggests that what we thought about how cells know what to make is incomplete. And this is a way to sort of finish that story, or take a new road for that story.
If it holds that the these bioelectrical signals are controlling gene expression, or the patterns of where genes are expressed, we have a whole new approach to correcting birth defects or preventing them or spotting them before they happen. Plus, we have all these highly specific, beautiful drugs that have been developed in neuroscience or in psychiatry. We have all these drugs that we know work and are safe in humans that we can use to control electrical signals. And so the most exciting thought is that this could be translated into highly specific and powerful medicine to interrupt when normal processes go wrong.
Whatever the question is, drugs are always the answer.
Sorry, I have some questions before you go ahead.
Please.
So suddenly you changed the subject to others. I have one question for protein and adenosine. Once I explained to the adenosine, and you and we are the proventricular and the SVT can affect. We are using for this adenosine to then open to the vessel. But how you can make the relationship with the adenosine to the acupuncture to working process? Suddenly you just stopped, you jumping to the other subject. I'm sorry, it's my case. I didn't quite understand once you explained the protein and adenosine, how you can make to then the biochemistry can impact for the acupuncture. Because too many chemistry is explained. Can you really be more explain to adenosine how they can relate it with acupuncture with some the movement is for using to the chemical part with the material, you know?
I'm naively optimistic that your question will hopefully be answered as we continue. If not, I apologize in advance. Yes, that is the intention. We'll see if we succeed, and I will make sure to circle back if we don't. So, just to show this again, in light of that video, so what we were seeing with that video was a frog embryo where everything was smooth, and then you could see those electrical signals come, and then you had a frog face. So again, there's an interplay between this, what did she say, this kind of this energy that seems to control everything in the body, you know, who would have thought, and then this sort of the biochemistry of it, right? So all chemistry has an electrical component, and so we see where we see these concentrations is going to influence where things show up in your life.
We're able
Sorry, guys. Yeah. Okay. And so, you know, again, Michael Levin, and this is, you know, a little bit technical, but just to kind of give an idea of what he's proposing, and I don't know what his relationship is with acupuncture in that system. I would be surprised if he wasn't sympathetic based on what he's proposing. But again, that we have things like the environment, and we have genes, and then we have these morphogens, which is this chemical gradient. Then we have the bioelectrical signaling the electrical dynamics, and then we get a chemical. That seems to be what he's saying. And so, you know, so this theory that I kind of very awkwardly jumped onto without a smooth transition, isn't it exciting though? It was proposed by Dr. Charles Sheng out of Harvard. And so he refers to this as the growth control theory of acupuncture. And he writes, the growth control model is the only published model in basic acupuncture research which has met the gold standard. It encompasses the neurophysiology model and suggests that a macroscopic growth control system, like, you know, the pattern of the person that we observe, you know, in our clinic, originates from a network of organizers in embryogenesis. The activity of the growth control is important in the formation, maintenance, and regulation of all the physiological systems. several phenomena, and I apologize, I do try not to put so many words on my slides and then read them, but I feel like this is quite important. I feel like he said it well. So if you'll indulge me, do a little slide reading, and then I'll get back to confusing the audience. Several phenomena of acupuncture, such as the distribution of auricular acupuncture points, the long-term effects of acupuncture, and the effect of multimodal, non-specific stimulation at acupuncture points, so that you can press it, you can needle it, you can heat it, you can stretch it, are consistent with the growth control model. The following predictions of the growth control model have been independently confirmed by research results in both acupuncture and conventional biomedical sciences, which is helpful. So these are the things that make this theory kind of compelling, gold standard, and proven, according to Dr. Shang. 1, acupuncture has extensive growth control effects. 2, singular point and separatrix exist. So the separatrix is it's a mathematical concept, and it just means like it's a point where things change. Yeah. Exists in morphogenesis. Organizers have high electric conductance, high current density, and high density of gap junctions. A high density of gap junctions is distributed as these these separating points or boundaries at body surface after early embryogenesis. Many acupuncture points are located at transition points or boundaries between different body domains or muscles, coinciding with the connective tissue plane. So if we think about where the points are on the body, if you kind of close your eyes and picture Acuman or Acuwomen, depending on which way you swing, most of the points are at areas of the body with high levels of change, particularly the ear, because the ear really doesn't have to do that much, right? Sorry, ears. I like mine, but it's like, you hear and there is a nerve there, but like it's got over 100 points. Well, look at all the curves and all the changes and all the anatomy on it, right? So it's that quality that allows the ear to be so ear-like, and at the same time, the reason why it has so many acupuncture points. And some morphogens and organizers continue to function after embryogenesis. So it's a system that turns a single fertilized egg into a three-dimensional being, and it doesn't go away. It still helps with regulation. So, yes, please.
Morphogens and organizers, are they specifically known chemicals, or are they just kind of positive?
Yeah, they're specifically known chemicals. They'll come up a lot in your cancer research reading. One of them is, I'm not making this up, it's called Sonic Hedgehog. Right? How cool is that? So yeah, they do have names, and they are specific, which is great. And so, and part of the reason why I bring them up here is because then I wondered, so I thought, okay, well the best other theory about chemistry of acupuncture, besides pure energetic signaling, is Dr. Shang's growth control models. And I'm like, well, are purines morphogens? Do they play this role in this three-dimensional patterning? And it would be a very sad and weird lecture if the answer were no, but the good news is the answer seems to be yes. So this is a paper from 1992. That says extracellular purine nucleosides stimulate cell division and morphogenesis, pathological and physiological implications. And they write, extracellular purine nucleosides and nucleotides are ubiquitous, phylogenetically ancient, intracellular signal. I love the way that they write about these things. Researchers do not seem to get quite as excited about other types of chemicals. Purines are released from hypoxic, so tissue that doesn't have enough oxygen, damaged and dying cells. And so here they're talking more about the role in regeneration, right? We're not talking about embryology in this particular instance. Purine nucleosides and nucleotides are potent mitogens, so like causing cells to divide, for several types of cells such as fibroblasts, endothelial cells, and neuroglia, right, brain cells. We propose that after injury to tissues, including the central nervous system, purine nucleosides and nucleotides interact synergistically with other growth factors like BDNF. They stimulate proliferation and morphological changes in the various cell types involved in the wound healing response. Okay, so and again, that's why I believe that's why we have the embryology system is that the same system that allows us to heal throughout life. And I would also, and this is definitely for discussion, I would say that cancer is embryology gone wrong. that same system again. And then we see in 2012, purines as potential morphogens during embryonic development. Yay, we come full circle. So here we see just the kind of different phases of embryogenesis of the mouse, a chick, that xenophus again, which is kind of cute, and the zebrafish. And then here, what they've done is they've mapped where these different adenosine and ATP receptors come online. What they found is quite a bit of redundancy in the system. So when they knock it out or they remove anything, often it only results in a fairly small change, except if you get rid of the adenosine receptors, then you don't have a viable embryo. So that seems to be rather important. So, in summary, before we get into a riveting discussion, and again, I want to summarize this because if you kind of, if what I described resonates with you, okay, so if what I'm proposing as a central and kind of particularly important biochemical basis for acupuncture signaling, if you're buying it, then this is a very kind of clear, concise way for you to internalize it and make it your own, so that if someone says, nobody knows how acupuncture works, it doesn't work, it's just chi and fairy dust, you can say, well, actually, acupuncture needling triggers the release of ATP and adenosine, and these bind to nerve receptors that send the signal from the spinal cord to the brain, and that in the brain stem, this regulates all sorts of functions that we claim that we can regulate, right? And that in the higher brain centers, this modulates pain. Oops. And that ATP and adenosine really seem to be the starting domino for quite a lot of different chemical processes, that when you stop that step, things don't tend to go forward. And if you start other steps, it doesn't seem to have the same impact. And that this explanation removes doubts about biological plausibility of needling people and doing amazing things. And it explains how acupuncture can be clinically helpful for such a diverse range of clinical areas. And then what we can now add is that acupuncture points and channels coincide with morphogenic organizers, and that purines, including ATP and adenosine, are morphogens. So that is one way to explain how acupuncture works. So I kind of, at this point, want to open it up to some discussion. First, maybe questions. Did that come around for you?
Yeah, so one thing, more than the different question. More than the adenosine, I mentioned the protein and the adenosine can chemical bio. For example, if many Korea and China, we are using the pharma acupuncture, which is including to the biochemistry material, put together and the acupuncture point directly to working well now. So, which is we have so many articles which is the function and more effective for rather than the manual acupuncture needle. In your lecture for today, you know that the adenosine and the protein, you really wanted to is working to then some acupuncture point with the circulation. Do you have any sort of evidence compared to pharma acupuncture with biochemistry material, including protein and adenosine? You understand
It sounds like you're injecting.
Yeah, or injecting for material, the adenosine and the protein, injected directly to the chemical.
And you're injecting adenosine.
Yeah.
Okay. So my talk wasn't too surprising. And you're asking me, so my brief answer is no, I have not seen any research comparing injecting adenosine to manually stimulating it. I would say, with respect, based on my reading of this, I can see, it would seem to me that manually stimulating the points might be potentially safer. Simply because of this, this feedback inhibition, and particularly the literature that I pointed to showing that it's really the balance and the relationship between adenosine and ATP that will stimulate tumor growth or not. So when you inject adenosine, I think it's possible that you'd be down-regulating ATP production, because that's how a lot of it works. So I'm not suggesting that I know that it's harmful, but personally, there seems to be advantage to using mechanical stimulus.
Well, that's right, and adenosine can using for people who had heart problem. That is using for the chemical part adenine very well on the medical Western medical part in that material, the protein and adenosine.
Yes, yeah, and I believe are they also using it for like analgesia as well, like for certain. Yes, so so great, and I think there's probably instances where it's appropriate and it's helpful, but if we could take the conversation a little bit wider and how I normally talk about acupuncture in general, is that we really want to start at treatments that have the best balance of effectiveness and safety, and move up the invasiveness scale as needed, right? So it makes sense to me, for example, to try acupuncture. If that does not work, by all means go have your knee operated on, but not to start with the surgery. So it makes sense. I'm aware that there are uses for it in medicine and that it gets used, but my first preference would be to stimulate improved self-regulation of the system before going in at the bottom, because I think you could have unintended consequences. That's my personal philosophy.
But in the summary that you mentioned, the acupuncture point and the channel and combined with other organizations, isn't it? You are summary then. You summary then that is
Summarize. Sorry, that.
acupuncture point and the channel and combined with and the menopogenic organization. You summarize it.
Yes.
This is the protein and the adenosine. Can you measure to the action to the with the organization? My understanding, the chemical, the biochemical, the material can impact for some acupuncture manual or the pharmacy, and it doesn't matter if it's working or not. I need to do what is your opinion, the biochemistry and approach? You can get that approval acupuncture? You can talk to them okay, the biochemistry is clearly the acupuncture needle and channel is working for well with the biochemistry the view. I need to then your opinion, not in the paper. Once you research it, I just want to do the summary. What is your opinion? The biochemistry can acupuncture point is working or not on science field?
I'm not entirely sure that I understand the question.
I'm sorry that.
So I apologize.
Yeah.
So I will thank you for your question, and I see that there's some other questions. Okay. Yeah.
In this sort of complex puzzle, does substance P have any relationship with this?
Yeah. So that slide that was earlier on that showed like all of the biochemicals that acupuncturists love to share on Facebook because it makes it look like acupuncturists do lots of things, I believe is one of those. Yes, so substance P is closely related with pain signaling, and I think it's going to be downstream again of ATP and adenosine, and I wonder if when we get chronic pain set up, we get this sort of lack of inhibition of the signal. It may be that the yin-yang balance of ATP and adenosine is not switching back, if that makes sense. I think it's at the beginning. Do you remember which one I'm talking about? No. Okay. Slide 74.
Can I just ask where we would get the slides?
You can email Kip.
So, wonderful. Thank you for that. There's actually a bit unclear. There's an unclear, not like a very essentially what you're saying is that the first response to the acupuncture stimulus is adenosine related.
The first chemical, yeah.
But then everything else that happens consequent to that whole neurological cascade that produces all the responses that we love so much are related to numerous other responses. So really you're talking here about specifically the mechanism that is the first response to stimulation.
Yes.
That's right, isn't it?
I believe so, yeah.
In a sense, that doesn't necessarily relate to morphogenesis or a whole load of stuff that you're talking about is kind of very interesting, but perhaps rather peripheral to the first response.
So the first response issue, so that's explaining how ATP and adenosine are the first chemical response initiated, that if we block that, pain relief doesn't seem to happen. But then we're still left with the question of, well, what does this have to do with the three-dimensional map? Right, and so what we find then is that not only are those chemicals initiating really a range of like where we have cells like fibroblasts or less differentiated cells, ATP and adenosine is triggering their differentiation for healing. That happens mostly along the channels. That happens more along channels than non-channels.
So you're saying that the adenosine system is also at the effector stage, at the end stage of the process as well?
It's triggering healing, which is the same system as the embryologic growth, and there are more fibroblasts. They're more densely populated. All these cells are everywhere, but they're more densely populated along channels than they are along non-channels.
So supposing we could look at needling stomach 36 and its effect on brain-derived neurotrophic factor here.
Yeah.
You're saying that the response there is also adenosine related, for example.
That's my understanding at this time.
Okay. Good.
Yeah. Yeah.
So you've got kind of a bit of a suggested kind of model for local effects, particularly if they're on these morphogenic lines.
Yeah.
And you've also got a suggested model for like general effects. So needling in a place which produces a lot of ATP or whatever is kind of going to affect lots of things in the system in a general way. But then, so the gap is like, well, let's see,
White
specificity can help?
Yes.
Can that help digestion?
Yeah, no, that's a good question.
And you
Yeah.
haven't got that.
No, I don't. And
you kind of got some suggestive stuff.
Yeah, I mean, I suspect that there are other things that play, like nerve reflexes. I suspect that if we look at the embryogenesis and how things fold, and that's been suggested by Dr. Cowan in his book Spark in the Machine, which was based mainly on Charles Shane's work with some of his own contributions. He's talking about how, the folding around the stomach organ kind of comes out through, the stomach channel, and so that there might be a physiological connection as well. So you're right, I don't actually have clear answers on that. It's, what seems to be more is that when we close our eyes and we know where our body is in three-dimensional space, that's not really mediated just by nerves, because the nervous system's looking for change. So if my hand is here and it stays here, there's not really, there's nothing to communicate, but it is the ATP and adenosine, it seems to me, that is providing the chemical basis for that nervous system and that three-dimensional map in the brain. So yes, there are still questions.
Do you think that the chi sensation is linked here? How would you describe that, when you really feel?
Yes, yeah, I do. And again, I don't, there's been some, because then I know that acupuncturists are a little bit heterogeneous in their views on the chi, so it's not, I'm not going to weigh in strongly what people do clinically. There are some studies that suggest that it's important for the clinical effects and for pain relief, and it seems to be related, and this is a lot of this comes from Helene Langevin work about connective tissue modeling. And so when we insert the needle and we rotate, they had like a little camera, and you see the loose collagen fibers starting to wind around the needle, and then I believe that once it gets to a point where they're attached to where they're attached, but now they're wound, you get a pull. And that seems to be related.
This is a definite thing, isn't it? We all felt it.
Yes, on the receiving end, on the giving end, you feel it. And as well, I'm trying to think if it was her work. It might have been that they introduced a chemical that would prevent that from happening, and the effect they've.
Bleeding would be quite interesting here, to looking at what bleeding does, just
Yeah.
removing of everything.
Yeah, no, absolutely. And again, I want to kind of circle back to how I framed this when I started, which is to say I do not believe this is the only explanation of how acupuncture works. I don't think it encompasses everything. I don't think we could even, we cannot explain everything through reductionist methods. This is simply a model that I think seems to me to be very important and very useful for a variety of reasons. So that's why I present it.
Just as a very practical question for my patients. I know a fair bit about caffeine and its interaction with antidepressant drugs because it sits on the receptors. So my question, I would go recommend to my patients now that they don't have caffeine before treatment. But what's the time?
Yeah,
The system.
that's a good question. There are a few studies that have been done, mainly on mice. And they show that it's dose dependent and time dependent. What I would add to that is your genetics, right? So I'm a slow CYP128 detoxifier, so caffeine stays in my system longer than someone else's. So, and I just, I don't want people to get their knickers in a twist. I don't think, we don't have enough clinical evidence to make a strong recommendation around this, so it is just a weak one, but it sort of makes sense if you can think of caffeine as both being a treatment for migraines and an inducer of migraines. It's this system that we're messing with. So I would say that it's a little bit unpredictable how caffeine is going to affect our needling. In some people, maybe it would enhance it. I just think it's an unknown entity and it's going to block adenosine. So try it and see.
Yeah, I understood a mechanism for acupuncture through fascial planes and some kind of electrical signaling in that way, through sort of a path of least resistance along structural fascial planes.
Mm-hmm.
Is it perhaps not so well researched because it's easier to study chemical processes than very subtle bioelectrical processes?
I think that's accurate, so I subscribe to that. I think so one thing about explaining things is the pragmatism. Who are we explaining it to, and what's their worldview? And conventional view of medicine is very chemistry dominant, which I don't actually think is for any particularly good reason. Then a lot of people are selling chemicals as solutions to problems, and so the electrical system of the body has been very under-attended to, even though if reading Michael Levin, it may be more important, and we certainly find it to be more important in our practices. So I think that's probably correct. It's probably studied a little bit less, but I only don't present it here because it's probably not going to be as convincing to your medical colleague, even if it's accurate and well-researched, because of their biases.
Thank you for the presentation. My question is, as you mentioned, at the moment, almost all these mechanisms or basic research are based on the animal explanation, most of them are mice. But one question, even during my PhD research, the defending, I did my research for acupuncture for the depression. We got a model of the mice. A question from the researcher said, how do we translate from an animal perspective for a human being? We use the same needle. For human being, it's a tiny needle. But for mice, it's like a big stick. For human being, we have some emotion involved, placebo involved. But for animals, they have not this kind of. But how do you translate from this animal expectation to our human being mechanism?
Yes.
Do you have any suggestion on this?
Well,
how to answer this question?
Yeah, so the positive news is that the study that I presented demonstrating that adenosine receptors were required for pain relief in mice was translated into humans. Yes, that is one study, but it is a human study, and it was a really well done one. So that is a partial solution. But then most of the research that I presented on the clinical relevance of adenosine and ATP was done in humans. So yes, actually one thing I wanted to mention, because that's a really good point, is that I had another theory. I was going for a jog yesterday after I realized that purines are morphogens. And then I was going for a jog, and one of the biggest inducers of BDNF, brain-derived neurotrophic factor, is exercise. So, you want to grow your brain, go for a run. But then I realized, well, so ATP is triggering the BDNF, so there must be a little bit of hypoxia. And then we get back to this idea of a hormetic stressor. Have people come across this idea of hormesis and hormetic stressor? So it's really summed up in the phrase, the dose makes the poison. So the conventional view of things is like, this is good, this is bad. Let's keep it simple, right? But we know that's not the case. And really, low level of something potentially bad in high doses is good in low doses. So exercise is a good example. You're causing yourself a stress to trigger a positive adaptation and a bit of hypoxia. Too much hypoxia is not very good for you. So with the mice, the way I would sort of pragmatically try to translate that and say, look, perhaps the scale means that it's 100 times thicker, but for this mouse it still seems to be beneficial. And we have that option in humans, but we choose to do the smaller version. But it did make me wonder if ATP and adenosine and parenergic signaling are actually the commonality for all hormetic stressors. So another example would be, we often think of a lot of fruits and vegetables as being antioxidants, but actually they're mainly mildly pro-oxidant, and they trigger our own body's antioxidant system to adapt. And so it may be that basic thing, that a little bit of oxygen deprivation or damage or mechanical stress or poking or heat is the first thing in that step of causing the body to adapt and heal and get stronger.
Which is how they've described how direct moxia works, because what it does in terms of denatured proteins and bringing it up, immune reaction, white blood cells.
Yes, that makes a lot of sense to me. And actually, I've recently come up with another theory, I think, in addition to that. So she was saying that that's one theory of how direct moxia might be working, that's causing a helpful stress to the local area to stimulate protein degranulation and bring immune cells. So, but recently, kind of I assure you this is not connected to the talk, so please don't get confused. There's a book that came out a few years ago highlighting a theory called the fourth phase water, which I actually feel might be very relevant to how to the Chinese medical model of how the human body works. And so the researcher outlines that in addition to the three phases that we know and love and are familiar with, ice, liquid water, and vapor, there's a fourth phase that appears when water is next to an electrically charged membrane. And what happens is, whereas normal thermodynamics would anticipate that maybe you'd get a tiny little bit of ordering next to the membrane, then the rest of it would just be random molecules having a boogie. But that what actually happens is for hundreds of molecules, you get an ordering, and it's positively charged, I believe. And the ordering is in a hexagonal structure like ice, and it excludes all solutes. And then after that, you have the corresponding negative area, and what you have is like a battery. And that battery gets charged by the sun, and it gets charged by light, and the best frequency of light for charging it is infrared. And it seems that moxa, in addition to giving off heat, gives off infrared and may be charging that battery. And my next thought, which I wondered, and this is genuinely a question, not an answer, and if anyone does have thoughts on this or more expertise, I would welcome it. I started to wonder if that might not be a helpful explanation as the corresponding overlap for the Sanjiao, because we have these kind of larger membranes, so we might have a larger kind of group of fourth phase water in this structure, and then you have this organ that doesn't have a form, that maybe this is what's been referred to. So I don't know. So it's definitely interesting for further thoughts. So, does that answer anyone's question or anything like that?
The next question is about the channels, the points in molecules. As we know now, one of the critical problems in Western economic research are economic working. But whatever you put needles,
yeah,
the needle is a placebo essential. But there are some research indicate they treating some heart problem, they put it on different points on different channel. It may generate different results. You may know. How do you understand this from this?
Problems with point specificity. Yeah. So the question is, how do you answer criticism that it doesn't matter where you put the needles? And there's a lot of people who work on that problem. So it's a multi-pronged answer. Part of it is if you look at the literature, the acupuncture treatment isn't that good, and there's often not enough of it. And then the sham, what we tend to find in research, and Mark can correct me if I'm misspeaking, is that studies that give more treatments tend to find a difference. So for migraines in the Koppelman systematic review, I believe there was the cutoff, they were looking at 12 treatments or fewer compared to 12 treatments or more. And 12 treatments or fewer did have a small difference between acupuncture and sham, but if you then do 12 treatments or more, it was like a 0.6 effect size. It was like more than moderate. So part of the problem is control, and part of the problem is the treatment, and inadequate dosing, just poor design. So I think the way I would answer that is by focusing on dosing, because that's a concept that most people are familiar with. If you give someone, not enough. Well, of course, we went over yesterday that Tylenol doesn't work anyways. But if Tylenol or paracetamol did work, and you only gave a little bit of it, then it would be closer to a sugar pill. But if you gave more of it, then its differentiating effects would be greater.
So two short questions. And the first one is, and it follows on from a couple of questions here, which is when we're thinking about things that have no understandable point of survival, where we're having a secondary reaction. So let's say there's some sort of, we can affect motility in the bowel through that. So what are your thoughts on those aspects of the choice? And then the second one is, what are your thoughts on non-specific or like the long-term effects of acupuncture in relation to that, I don't know, attractor force regarding growth, growth theory you just spoke about?
Yeah. So the first question was on regulating functions that have no survival benefit.
Yeah.
But pooping's kind of important.
Yeah, no, but I mean by stimulating those channels.
Yeah. I don't. Tell me if this does answer your question, but I think part of the problem that we've had is that we learned a system of how the human body works that doesn't correspond to the conventional model, but it's very unlikely that acupuncture exists for us so that we can be acupuncturists to needle them. And so that's where the growth control theory is particularly helpful, because it's shown it doesn't exist for us to needle them. That's just a pleasant side effect.
Yeah, I understand that from an immuno perspective or circulatory perspective,
yeah.
But for something that's specific to, I say, gastric.
Well, it's just my understanding is that that ATP adenosine is the signal that is how people know if their bowels are full or empty. Is that ATP and adenosine as a cofactor has bound to give that information back. And again, I think pooping is helpful. So I might have misunderstood the question. And then the second question was about the long-term effects. Yeah, so I think what Charles Shang was positing, and that wouldn't have occurred to me, but I can see what he's saying now, is that by helping to self-regulate at the level of this growth control, patterning is how we're getting long-term effects, because perhaps those changes last. So it's not just, if you put a chemical into the system, then once it's metabolized, the chemical is gone. But if you put input in, some sort of input mechanical stress at a certain place, it communicates with the whole three-dimensional organization, and that change stays. So changes in the rate of the fibroblasts being changed into skin, which is to say healing, is going to increase because the signal for healing is increased, which is maybe part of the distinction as well about injecting adenosine versus stimulating it. I feel like I've criticized this whole treatment, which I didn't mean to do. I saw some more questions. In the back.
I think your idea is brilliant. I think there is some kind of connection between your idea and some other knowledge. I just want to ask a short question. Because this material, in a sense, is related with lack of oxygen. So, and this can be converted back into ATP very, very quickly, probably to do it. So, according to the existing evidence, when we perform acupuncture, it is because the needle actually tweaks the cell release the existing one, or it is because the needle gives the stress or pressure over the cell then converts ATP into it. Is there any evidence about this?
Yeah, that's
Is it just simply purely released, or it is converting ATP?
Yeah, so my instinct is that the answer is that it's likely both. But one bit of evidence that we can find comes from the lovely picture of this happening.
Yeah, I remember you have a picture of a needle and the surrounding chemicals.
Yes.
Yeah.
Yeah, so in this picture, you see it poking a cell, rupturing the cell. But what happens, and that may happen with acupuncture needling, so I suspect that's part of it. But I think very specifically when we insert and then we twist and then we get loose collagen tissues winding around the needle, that deformation triggers fibroblasts to degranulate.
Okay.
And that's I think that's the bigger part of the signal.
It's the physical stress causing
It's the physical stress causing deformation and then causing the fibroblasts to degranulate, and that's I think the bigger part of the signal.
My question related to that is, if the person is just have done a workout or a good exercise,
Yeah.
with a exhaustive feeling into that acupuncture training room, because they are already using a lot of energy to be converted into this one, will this reduce the acupuncture effect because the existing in the tissue, or we haven't got any evidence?
Yeah, so his question was about treating someone who's just been doing vigorous exercise, and so they might have already been stressing their system and maybe using up energy, you know, I agree. So again, there's more factors at play here, so I think it's difficult to isolate the two. Just on the top of my head, I would say probably just after exercise may not be the best time for like a regulating, balancing treatment, because you're giving two different hormetic stressors. So I don't know. So you know, you're training the body that needs to get better at something, but you're like trying to, I don't know, like learn math and physics and you know, Spanish at the same time. That might be a little bit harder, but then, you know, if my patient's exercising before acupuncture is the biggest problem that I have, then I'm quite happy with that.
Thank you very much.
Yeah, thank you.
Do you have any sense of how this system of kind of morphogenic response or adenosine explains the difference in effective, say, stomach 44 as opposed to stomach 36? Is there a sense of how that was?
Yeah, so, you know, again, I think, you know, part of the answer to point specificity, we can look at nerves, and I hope everyone in this room is familiar with the work of Pony Chang. He's based in Toronto. He has been doing extensive cadaver studies, and he's fluent in ancient Chinese, so all kind of helpful things. So what he is showing is that each point is located at a nerve, actually, I think he's focusing on the nerve aspect, but a neurovascular bifurcation. So what he does, which is very clever and very spooky and fun to watch, is he will take a volunteer and he'll, you know, there are points that, in my mind, because I'm kind of a lazy acupuncturist, so I don't differentiate, you know, the different effects of these points that might be very close together. Let's say it's, you know, 736 and 735. Put the point in with the needle stimulator, and it'll show you the different nerve classes being activated. So that's going to be a component of it.
It's a kind of combination of different systems.
Yeah, I mean, my personal feeling on this, and it's always my dream to get some of these people together in a room so that we can kind of like nerd it out. So, you know, Pony Chang, I believe, I hope I'm not putting words in his mouth, thinks that the points are those bifurcations. I think that bifurcations exist because of the channels. I think that you have electrical energy and you have morphogenic gradients that create the signal for the bifurcation, and the points are in spaces. and maybe not an important point. But then also, if we go back to the morphogenic, the growth control model, and to me this made the acupuncture points make so much sense. If you think about the acupuncture points being more common in areas of change, because the question that the central dogma of biochemistry does not answer is how does my arm know to put an elbow there? And that's where a lot of points are. And that's where we have different bones and that sort of thing. So part of the differences in those points are going to be that it's just a bit further on. It just has a bit less density of those morphogens.
But the same morphogens were just in less dense.
Right, same morphogens, same taste, less filling. Same morphogens, less dense. No.
So
yeah.
Which is, and again, I know it's a fairly sort of out there theory, it's hetero and has been discredited, but what are your thoughts on Rupert Sheldrake's morphic resonance theories?
You're going to get me thrown out of here. Makes sense to me. So you say it's been discredited. It depends on it. So for those who watched the talk yesterday that I did on the different levels of consciousness, that's a yellow model. So what I'm babbling on about there is that we talk about what science shows, but I actually truly believe that there are multiple sciences. So the dominant science at the moment is materialism. And you can't, if you're looking at a non-materialist idea, it's always going to look wrong through the lens of materialism. So you can discredit that easy peasy, just like you can discredit practitioner-patient relationship by looking at chemicals. Or, if you look through that lens. So, yeah, it makes sense to me. Yeah, I'm looking for a morphogenic gradient. I think I just saw a couple more questions.
Could you explain the biochemical reason behind the effectiveness of cosmetic acupuncture? Can you explain to guys how it works?
Yes.
Yeah,
absolutely. So I would mention collagen synthesis. So, and again, we're going back to that idea of a hormetic stressor. So if you punch them in the face, that probably there'd be a larger stimulus, but would not have the desired effect. You can do but see, I'm probably not very good at selling cosmetic acupuncture, so don't use my you can explain it. It's a tiny signal, like microscopic exercise. That's better. Than a tiny punch in the face. And then that is going to give a signal to stimulate collagen synthesis. Yeah. Yeah.
I just read that the biggest sources of ATP it's actually meat and fish. Yeah.
Yep.
And the other sources is like nuts and vegetables, providing that so many people going vegans or vegetarians, completely excluding meat, fish. Do you actually can see any difference in the sources of animal origin or vegetarian origin?
So that's a good question. So she was asking about how she kind of just looked and saw that the largest dietary sources of ATP and adenosine and purines are coming from animal sources, which makes sense. In fact, people with gout are told to go on a low purine diet. So the question was, do we see a kind of difference in this reaction between vegetarian and I think that this is sort of explained a little bit in our own theories about if you're going to use just acupuncture in someone who's very blood deficient and what results we might expect. So I think there's something to that, and I also that I wondered. We remember what morphogens look like. I'll let that go. That I wondered if the effectiveness of acupuncture might be a little bit dependent on someone's protein status. So in my practice, in addition to doing acupuncture, I do a lot of functional testing, and I can see on an organic acids test, the urine test, that some people are very low in some of their amino acids, and that not all of them are vegetarians coming from gut dysfunction. So it goes back to everything starting in the gut. So yes, I think that there might be an issue, whether it's through inadequate intake or inadequate digestion and absorption. that acupuncture is a regulating treatment. It's allowing you to work with what's there. So the same principles for whether you're going to use an external versus an internal or a combination apply, and this is maybe another way of thinking about it.
What are your thoughts on fasting and an amazing effect of the body at a DNA repair, a so-called ketogenic way of living, and acupuncture and supporting the ATP?
Yeah, that's a really great question. So I think of fasting as being like a hormetic stressor. So I actually get a little bit concerned because just like exercise is good, we're not going to all take any patient and have them start running a marathon. I think a lot of people do need a certain baseline level of metabolic fitness for it to have a beneficial effect. So I do worry that I think that fasting, when used appropriately, can be very powerful. And I think it's in line with how we evolved. But that some people might be introducing it at the wrong time. And actually, I was mentioning to Harriet earlier, and this is totally unrandom and totally biased, just what I observed in terms of acupuncture effects. We're saying that when patients come in, you don't know if they're going to, how much benefit they're going to get. Is it going to take 1 session, 12 sessions? I said the pattern that I see is people who exercise get fast results with acupuncture, and I feel like they are just, and so that maybe someone's exercising but the diet's not good and they're not mindful, versus somebody who's sedentary but maybe they're very mindful and they're eating well. The person who exercises seems to just really get quick results from acupuncture. It seems to me that they are like priming that entire system through their movement. I think there's a piezoelectric effect as well, that we're generating electricity through impact, and that means that this tool, so going back to your exercise question, that is helpful but maybe not just for. Okay. If there are any other questions, feel free to approach me afterwards. But thank you so much for